I'm Deanne Erdman with Baylor College of Medicine's Options Program, and this presentation is a review for the Life Science Teacher Certification exam. In this portion that I would like to look at today, we're going to deal with biological classification. So the first question becomes, why classify organisms? When you look at the amazing array of organisms out there, everything from microscopic to bacteria to giant redwoods, yeast, jellyfish, zebras, and the list goes on, it becomes pretty clear that unless you have some sort of an organized framework, you're not going to make any sense out of all of this. So we want an organized way of looking at this. We want to analyze the patterns of development and in some way try to establish and reveal what the evolutionary relationships are. So another question comes, why does it keep changing? And of course that is because of our expanding knowledge. And with that, I think it's important for you to remember that while when you were in school you might have had to memorize a lot of classification, you find that a lot of it's obsolete now. So it's much better for students to learn to critically think, to use classification systems and understand how they work, not just to memorize them. Taxonomy is the study of naming and classifying organisms into some sort of diverse groups. And I'd like to explore this with three major topics in the next slide. The first being binomial nomenclature, which is a two-word naming system. Hierarchical classification that deals with 7 classification categories, beginning with kingdom and ending with genus and species. And systematics, which can be thought of as the study of biological diversity and how that diversity evolved. Let's start with binomial nomenclature. And of course, a Swedish biologist named Carolus Linnaeus first proposed this system, and it really replaced an archaic way of describing organisms. Organisms up until this time were described in phrases of 12 or more words. And you can imagine just passing that along from person to person, and then the complication of interpreting that from language to language. It was subject to just many miscommunications and misunderstandings. And so this two-word name assigned to organisms that Linnaeus proposed really has proven to be a very workable system that we still use today. In Linnaeus' time, scholars wrote in Latin, and we continue that process. So organisms are assigned a two-word name, genus and species. The genus is always first. It's a noun, it's capitalized, and it's underlined or italicized. The species is descriptive, it's lowercase, and it's underlined or italicized. And just a quick example of a binomial name for humans would be Homo sapien. Homo meaning man, sapien meaning wise. Linnaeus also, in a similar work, described a hierarchical classification system using 7 taxa, beginning with kingdom, then phylum, class, order, family, genus, and species. And sometimes you may be asked on a standardized test to work with this classification scheme, and you might get a little confused. Does class come before order? And a very useful mnemonic that you can use with your students as well, where the first letter of each of the words in the mnemonic help to spark your memory about the taxa, is King Philip came over for green soup. When you're looking at these classification categories, each successive level becomes more and more specific. And for example, if I were talking about bears, all species of bears are mammals, but not all mammals are bears. In the 18th century, organisms were divided into 2 major kingdoms, the plant kingdom and the animal kingdom. And this persisted for a long period of time. And even with the discovery of microorganisms, different things were fit into one of these 2 kingdoms. Bacteria were grouped with the plants because of their cell wall construction. Fungus were grouped with plants because of their cell wall construction and the fact that they were non-motile. And then the microorganisms that were able to move about and not photosynthesize were classified with animals. In 1969, Robert Whittaker proposed or identified two fundamental differences in cell structure, the prokaryote cells and the eukaryote cells, and with that proposed a 5-kingdom system which has been widely used. And as you take a look at the chart, you'll notice that in the traditional 5-kingdom system we have the monarans, the protistans, plants, the fungus, and the animals. With recent work with nucleotide sequences, ribosomal RNA, and different kinds of proteins, there is some mounting evidence that the lineages of the prokaryotes is very, very different. And so you can see several other proposals, a 3-domain system and a 6-kingdom system. And I think that this is an area where you will see a lot of other changes with the rapid advance of molecular biology, particularly in the protist group, which is sort of a catch-all category for anything that doesn't fit in the other groups. But once again, learn to look at and identify these types of systems, and not so much to memorize them, but to work within them as a framework. Systematics is an evolutionary classification of organisms, and to understand this classification from an evolutionary point of view, systematicists use or gain information from a number of areas. One of the first is fossils, and fossils are remnants or impressions of organisms which have lived in the past, and these fossils are gathered worldwide to establish a geologic timescale to better understand the succession of organisms. Homologous structures are another area that systematicists use, and one of the things you have to be careful about in looking at homologous structures is not to confuse them with structures that are really analogous. An example of an analogous analogous structures might be if we were comparing the wing of a butterfly and the wing of a bat. While they have the same function, they're very different and don't indicate any sort of close ancestry. On the other hand, if we were to look at the arm of a human, the flipper of a whale, the wing of a bat, and say the front limb of a horse, we would find that there's the humerus, the radius, ulna, carpals, metacarpals, and phalanges, all similar structures but adapted for very different functions. And this does indicate a more common or recent ancestry. We find kind of a similar pattern when we look at vertebrate embryos as we follow that development as well. So homologous structures are a useful tool to systematicists. Cladistics is another important tool, and it's used to show hypothesized relationships between different groups. And it's based on the idea that members of a group share a common evolutionary history. In cladistics, you use a derived characteristics, which are unique shared features. Another area that we need to look at, comparison of DNA and RNA sequences among organisms, and they reveal very important relationships. And we're particularly interested in the ribosomal RNA, chloroplast DNA, and mitochondrial DNA. And finally, molecular clocks, which is somewhat controversial but very interesting, and this compares the rate of change of nucleotide sequences in macromolecules. So as you look at this list, you begin to understand why we are starting to see so many changes in classification, because our tools we're gaining in the number of tools that we can use as well. As you take a look at these two diagrams on the slide, you're going to see, first of all, that each are similar in that they represent the hypothesis about evolutionary history. The phylogeny on the left shows a variety of evidences, or uses a variety of evidences, including fossils, and it represents hypothesized evolutionary relationships. The cladogram on the right differs in that it's based only on characteristics found in existing species, and each branch represents a new, unique, shared feature called a derived characteristic. The last thing that I'd like to talk about in this particular section on classification is the use of dichotomous keys to identify organisms. And a common mistake that people make when they're looking at dichotomous keys is to assume that they're the same as evolutionary classification. Dichotomous keys are just a useful way for us to find out what the name of an organism is that we're looking at or that we're investigating. So even though dichotomous keys use pairs of contrasting statements, they don't indicate evolutionary relationships. So when you look at a dichotomous key, and there's a small sample, you're going to establish pairs of contrasting statements, and at the end of each statement there's going to be a direction to go to a next statement to decide on, or it will end by giving you the name of the organism. In the example, it asks you, is the leaf simple or is the leaf a compound? And in each case it directs you what to do next, what step to go to. In step number 2, it asks about the margins of the leaf. Are they jagged or are they smooth? And you would continue this process until you end up with the name of an organism. Now, an incorrect pair of statements would be something where you compared, is the leaf simple and are the veins netted? So in pairs of statements, you need to stay with the same characteristic. And you can find opportunities to practice and use dichotomous keys in a number of different situations, not just when you're exploring taxonomy, but you might incorporate them in when you're studying snakes or when you're studying fish or other kinds of organisms, and give students a lot of practice in using these kinds of keys. This concludes a brief review of biological classification, and you can find additional information in the notes on the bottom of the slide, and also in the expanded content discussions if you feel like you need some more information. Thank you.